White pine was such an economic driver. Starting in the early 1900s, timber industry was well aware of what a good wood it was. It was perfectly suited for all kinds of purposes. And by 1900, the eastern coast white pine forest had been pretty well cut over, and so they had moved on to Midwestern white pine in Minnesota, Michigan. The Lake States had eastern white pine, and same big, huge stands of eastern white pine. And so those were being liquidated. And then people found out, oh my gosh, in northern Idaho, Washington, and Montana, in the moist forest there, they had western white pine, which was even bigger than eastern white pine, growing in tremendous stands. And you could have western white pine trees that were 2 and 3 feet in diameter, only 10 feet apart, which no other tree species can do that. So that meant what we call the carrying capacity on an acre was like 50,000 board feet of lumber per acre, and most other species it's a fifth of that. When they realized that there was so many white pine and it was such a good species, mills sprung up all over.
One of those mills was Potlatch. Built in 1904 by the Potlatch Lumber Company, it was the largest white pine mill in the world. Potlatch Lumber Company believed that in order to sustain its product, it needed to build a town alongside the mill. So by 1906, the company had built 128 houses, 2 boarding houses, 2 schools, a hotel, 2 churches, a store, bank, post office, and an opera house.
I saw trucks lined up through Potlatch, from the Potlatch mill, probably 40 to 50 of them lined up trying to get unloaded right up through 6th Street in town. But town used to be quite a little bit larger. We had a bigger school. In town, we had the Potlatch Hotel that was open. The hotel was a 3-story building. It had a barber shop in it. It had a restaurant in it. We had some dances. We had the Grange halls. The Granges around here were very active. That's where you used to go to fraternize with people. On a Saturday night, you'd have quite a party downtown because a lot of the loggers, and the loggers were tough fellers, every one of them. And, well, I was one of the halfway tough ones. There's not as many people around. People have moved to where jobs are, and that's what we see. The small towns keep getting a little smaller. The big towns keep getting a little bigger. But at the same time, we got a better lifestyle than anybody in the big towns.
Due to a declining lumber market, in 1983, the Potlatch mill permanently closed. But in the years before it closed, there were a series of events that resulted in the western white pine being nearly wiped off the landscape.
You summarize the white pine story and why it should be told. What's going on with this tree?
The white pine is the state tree of Idaho, and that should tell us something about why they're special to this state. They used to be a centerpiece in the economy of the state of Idaho 100 years ago. But now the old trees are almost gone. They have been logged, which is, a part of our economy. It's a part of our history. But there have been waves of diseases that have wiped out the trees as well. And now we're in a small patch of old growth forest. And even the top of this tree looked like it died once upon a time and was regrown. We think that it might have died from blister rust and then grown a new top. So it's hard to find in what used to be a landscape of ancient, massive trees. It's hard to find those trees nowadays. The state's record or champion white pine was called the Floodwood Giant. It was 230 feet tall. And we were going to climb that tree until we found out that it had died. It had died from blister rust, from one of these introduced diseases that's not even native to the United States.
White pine blister rust, it's a non-native fungal disease. It originates in Asia or Eurasia, made its way to Europe. If foresters in the late 1800s, early 1900s wanted to grow seedlings, what they would do is they would collect seed in North America and send it to Europe, France or Germany typically, grow the seeds out there, and then ship the seedlings back.
It's a fungal disease that can have up to 5 spore stages. But in order to complete its complex life cycle, it has to go between its primary host, which are white pines, any 5-needle pines, and then it goes to its alternate host, which are gooseberries and currants. They then go through other stages of their life on those plants, and then the spores at the final stage on their alternate host can then reinfect the pine. When white pine blister rust infects the branch, it comes in through a needle, and then it starts into a branch, and then it starts forming fungal tissue, killing tree tissue underneath the bark. And so what you'll see on a branch is you'll see swelling because that fungal tissue is growing underneath the bark. And then the bark will crack and the fungus will produce these pustules they look kind of like blisters. The canker will get bigger and bigger, and then eventually when it gets on the main trunk of the tree, that's when you have to really be concerned. Once that canker spreads and it continues all the way around the bole of the tree, then it's effectively girdling it, and everything above that is going to die, the same way that it does on a branch.
When white pine blister rust was introduced, people really quickly realized that it was going to be a problem, and it only took a few decades for it to really start wreaking havoc.
When we realized that ribes or gooseberries were the weak link in it, we were going to do everything to get rid of it. So the idea was, well, let's pull it out, or let's spray it. And actually, from 1910 about till 1945, we tried the eradication program. And they ended up with hiring people to go out, and they called them blister rust camps, BRC.
By about 1934, there was 11,000 people working the stands to remove every alternative host, the currant or the gooseberry. And they would literally create huge clear cuts, burn them as hot as they could, and then they would line up men between lines, and they would go and pick every gooseberry out of the system.
The spores that are produced on the ribes plants can't go very far. So they thought, oh, well, if we could get rid of the ribes, then the pines would be fine. And the pines produce spores that can go miles, but at that time they didn't realize how far they could go to find a ribes plant.
Ultimately, tearing out the gooseberry and currant plants did nothing to stop the spread of blister rust.
So what they decided to do was to give up on the wild white pine. So they quit favoring it in the thinnings. They quit completely managing it. We decided to go for a mass salvage. We took every timber-sized tree off this forest, as many as we could reach, and did that in about 10 years.
What they didn't understand was that some trees could get blister rust, survive for a long time with blister rust, and in fact be relatively healthy. So that was knowledge that we came to later. But that was after many of these stands were already gone. So what we missed was the opportunity to have that genetic diversity.
It was kind of a product of policy at the time, but I don't think that we should necessarily blame them because they were working within the confines of the body of knowledge of forestry and ecology at that time. We have learned a lot in the last 70 years, and with the benefit of all that knowledge and hindsight, now we would make different decisions, I think. Also, I don't necessarily think it's productive to cast blame or blame anyone for the salvage effort.
Fortunately, in the midst of the mass salvage, a man by the name of Dick Bingham began to notice something interesting happening amongst the stands of western white pine trees.
He was a very early proponent of seeking out and finding genetic resistance to white pine blister rust in western white pine. And he realized that there was genetic resistance at a very low level, but that it could be It could be captured and then utilized to produce rust-resistant stock for planting.
You would walk into a particular place and 99.999% of the trees were dead by blister rust, but there was this one, and it was fully crowned, had huge cones in it. It was just beautiful. And so he started scratching his head, and he started to realize that there's something going on with these trees.
Foresters would travel out in the woods and find a tree that was in the sea of dying white pine that showed resistance. And they identified over 4,000 of those coming from the Clearwater all the way up into British Columbia. So those trees have been used for a breeding program.
These are mostly seedlings from crosses. And these are seedlings that fared well in artificial inoculation trials that the Forest Service conducted for many, many years. And they didn't get infected, or they got infected and overcame the infection, and they were planted here for tree breeding.
Those trees have been providing the genetic material for the breeding program that's been in place for the last 75 years.
We're following on the shoulders of Richard Bingham, who started this whole program in 1956. The Coeur d'Alene Nursery in general provides tree seedlings for all of Region 1, which is all of Montana, all of Idaho down to about Riggins. We went back to stands where all the white pine was killed except for these few individuals. And we went to those trees. We collected cones, we grew the seedlings, purposely infected them with blister rust, and ran them through a 3-year trial in the field and measured all the attributes, measured when they were infected with blister rust, and recorded the ones that resisted blister rust. We also measured height growth on those trees, and so all of those individuals that were in the top percentages are now represented in our seed orchards. This year, we have orders for 500,000 white pine seedlings.
Today, all the western white pine seedlings planted in Idaho forests were bred to be blister rust resistant. That resistance comes in a variety of ways. One way is that resistant trees shed the needles before the fungus can enter the stem. Others wall off the bark at the base of the branch when the fungus reaches the trunk, so it can't enter the tree.
When people use the term genetically modified, it conjures up zombie trees, Frankenstein trees. I don't use that term because we've had agricultural breeding for hundreds of years.
All the genetics of that tree are in this package.
People have been breeding crops and improving them through standard breeding practices, and that's what we were doing. So yes, it is genetic modification in the sense of you're selecting for certain properties, you're selecting for certain traits, and you're breeding those trees together to increase the probability of those traits, to increase the numbers of trees that have those traits. But we weren't going in and, taking a little ratchet and tweaking the genes.
If you're going to plant a tree that's going to be there for 60 years, you probably want to plant a good one, right? Makes sense. Our focus is using what we call traditional plant breeding techniques and applying them to trees.
Research will inform industry decision on what to do, when to do it, and how it should be done. A 70-plus-year-old science experiment, like Dick Bingham's seed orchard, has had lasting effects, which is why the science must continue. Projects happening at the Priest River Experimental Forest are powerful examples of human ingenuity, patience, and passion.
For the spore trapping project, we have built these spore traps using a small motor assembly and a control board. They're built to capture spores in a way that not only verifies the presence of the pathogen that causes blister rust, but also potentially the abundance of the spore load within that area. And the traps do this by essentially having an articulated arm with a spinning motor on the end of it. And that motor spins at a set speed. We attach microscope slides to it. Those microscope slides are coated in grease. And as they spin, they capture whatever is in the air, particulates, spores, anything like that.
Then they extract DNA from the Vaseline solution. Ultimately, this is going to be used for a very precise and very targeted molecular assay that is only designed to pick up the DNA of Cronartium ribicula, which causes white pine blister rust. And we can use a whole bunch of mathematical equations to translate that to inoculum density in the air.
We hope to define a relationship between environmental conditions and topography and the infection risk on a site. And ultimately what we want to be able to do is be able to describe where you're likely to have a high infection risk and where you're likely to have less of an infection risk.
It does get me excited to come up with these sort of research questions, figure out how they're going to be implemented, how the experiment is going to be designed, what methods we're going to use, and then seeing the results. I mean, it's like opening presents on Christmas Day when you like make those first graphs, see what your results are, and like are able to like, oh, I wonder if this is showing the influence of X, Y, or Z on your results. And being able to show that to people, share that information with the world, that's all really exciting to me.
Can you describe what it looks like from up there and what the scenery and the environment of this area is?
We are in a small patch of old growth forest, and you can see the complexity of this forest. The trees are all different sizes, they're different shapes, and then out in the distance, as far as you can see, it's a rolling carpet of second growth forest until the farthest mountain range way out in the distance. And I mean, you can see tens of miles out into infinity. It's amazing.
For all their romanticized imagery of age and wisdom, if there's one thing we've learned, it's that big trees are constantly under attack. If a white pine happens to avoid being infected with blister rust, something else, in this case bugs, are waiting in the wings ready to feed.
Yeah, I believe this is an adult Dendroctonus ponderosi, or mountain pine beetle, that just crawled out of this gallery here on the inside of this western white pine bark.
It is a remarkably small beetle that has a remarkably large effect in forests. The adult female beetles chew into the tree through the bark, and they etch these galleries and lay their eggs in the phloem layer as they're eating through the phloem layer, or the sugary part of the tree underneath the bark. But the effect that this has on the tree, this disruption of that phloem layer, completely disrupts the physiological process of sugar transportation in the trees. The tree is effectively girdled and dies standing. So what we saw today was a relatively large western white pine tree that was completely what we call mass attacked by mountain pine beetle.
And yet, scientists maintain that planting white pine is the best way to ensure a healthy forest ecosystem, which is why, on a hot spring day in May, crews are navigating a hillside ravaged by fire and salvaged for timber to get white pine in the ground before the summer dry-up.
We were planting on a spacing grid of 11 foot by 11 foot, which translates to about 360 trees per acre. It's a 15-person crew, and on average, they'll usually get about 1,000 seedlings planted per person. Those men work very hard, and they're very efficient at what they do. The stands at the time of the fire were overwhelmingly stocked with western hemlock, grand fir, so we were primarily expecting to get natural regeneration of very large numbers of grand fir and western hemlocks, which is not in alignment with our forest plan goals and objectives. Our forest plan goals and objectives are pretty focused on trying to restore or return western white pine, western larch ponderosa pine, birch and aspen, cottonwood to the landscape again.
The best way to get it back into a healthy forest is take it all out, put the trees back in the ground that need to be there, that are going to be resistant to the disease that happen to be there.
White pine really does function like a keystone species in these North Idaho ecosystems, in the sense that it has profound effects on the entire forest community in terms of what you actually still have growing on that site. They have a more open structure that allows a lot of filtered sunlight to make it through even a fully stocked stand. So historic stands of white pine had this really rich understory community typically along with them, and all the plants and insects and birds that are associated with all of that. Well, in the wake of losing white pine, it didn't just leave gaps in the forest in perpetuity. There were other species that occupied that space, and predominantly the species that filled in the gaps as white pine was dying out were the more shade-tolerant species that would also grow underneath white pine. And the trees that have filled those gaps are grand fir, hemlock, and cedar. The way they grow needles, the density of the canopy that they grow in the layers of the canopy, means that they grab every last fleck of sunlight that comes down out of the open sky and leave very little light, if any, making it directly to the forest floor. And that inhibits any other plant growth. So much so that they'll even inhibit their own seedlings from growing. In terms of richness of plants and other animals and stuff available, those stands will almost become like a biological desert. When you get regeneration following a new stand replacing disturbance, you don't get white pine back, you get what you have. And so we've lost an enormous chunk of natural forest succession. These stands aren't able to remedy themselves because they don't have a seed source available on site to actually get there. So that creates a need for us to intervene somehow to try and insert white pine back on the sites.
Scientists are worried because the trees that have filled the space white pine left behind are particularly vulnerable to root disease, a tree-killing fungus that lives in the soil.
Yeah, we see the white mycelial fans of armillaria here. This is causing armillaria root disease in this western white pine.
Northern Idaho is kind of the root disease epicenter of the western part of the country. And what we see is that a lot of these species that have kind of filled in behind western white pine to take up its growing space as it's vanished, those trees also happen to be some of the most susceptible trees to root disease.
So what we're tending to see is this very slow, persistent mortality across the land, all over the place. And it's just, it's creeping, cruddy stuff. It's not the bright, flagrant kind of flamboyant stuff like mortality following a fire or mountain pine beetle. It's almost like a cancer. You can't see it, but it's there. And that impact is magnified because of the shift to species that are primary hosts for those root diseases. White pine is not a primary host for it, it's much more resistant to it.
We predict that root disease is going to kill in the next 8 to 10 years 25% of the forest. We're killing more trees with root disease right now than fires are. Our forest is falling apart because we just let the diseases out. We don't have the species diversity we had. And so we will see stands that are 50 to 70 years old dying right now. And we're losing the productivity. We have to attack the areas that have the root disease, that still have trees of economic value, so that we can harvest them and actually replace that and restore not only white pine, but western larch, ponderosa pine, so that not only do we end up with just white pine, but we end up with the 7 to 8 tree species that once were here. Our work is cut out for us. We really do need to move because the diseases are killing our trees quickly.